Cancer researchers play a vital role in advancing our understanding of cancer biology and discovering innovative treatments. Interviewers often seek professionals who are scientifically adept, detail-oriented, and passionate about oncology research. The following 30 interview questions and their 80-word answers are tailored to help candidates prepare effectively for roles in cancer research, whether in academic, clinical, or pharmaceutical settings. Use them as a guide to showcase your expertise, commitment, and contributions to the fight against cancer. 1. What inspired you to pursue a career in cancer research? My interest in cancer research began when a family member was diagnosed with cancer. This personal experience motivated me to understand the complexities of cancer biology and contribute to finding better treatments. During my academic journey, I developed a strong interest in molecular biology and oncology, which led me to pursue research that could make a tangible difference in patient outcomes and public health. 2. Can you explain your experience with molecular biology techniques? I have hands-on experience with various molecular biology techniques, such as PCR, Western blotting, RNA extraction, CRISPR gene editing, and immunohistochemistry. These techniques have been integral in my studies of gene expression and protein signaling in cancer cells. I've used them to investigate oncogene functions and validate targets for potential cancer therapies. I'm also familiar with data analysis tools used to interpret results from these experiments efficiently and accurately. 3. How do you stay current with the latest cancer research findings? I stay updated through peer-reviewed journals like Nature Cancer, Cancer Research, and the Journal of Clinical Oncology. I attend oncology conferences and webinars, subscribe to research newsletters, and participate in professional networks. I also regularly engage with research databases, such as PubMed and clinical trial repositories. Staying current ensures my research remains relevant and contributes to cutting-edge scientific advancements in the field. 4. What types of cancer have you studied or worked on? I have primarily worked on breast and colorectal cancer, focusing on the genetic and epigenetic mechanisms that drive tumor progression. My project involves studying tumor suppressor gene mutations and the role of microRNAs in cancer development. I've also collaborated on studies investigating targeted therapies in lung cancer, giving me a broader understanding of different cancer types and their unique biological behaviors. 5. Describe a challenging cancer research project you've handled. One of the most challenging projects involved identifying biomarkers for early-stage pancreatic cancer. The disease's asymptomatic nature made data collection and analysis complex. We had limited patient samples and needed to develop highly sensitive assays. Despite setbacks, I optimized our ELISA protocol, collaborated with pathologists, and presented our findings at a conference. This experience taught me resilience, innovation, and the importance of interdisciplinary collaboration in overcoming research challenges. 6. How do you handle unexpected results in experiments? Unexpected results are an integral part of scientific discovery. I approach them by first verifying the experiment's accuracy, rechecking reagents, protocols, and instruments. If the results are consistent, I explore alternative hypotheses and consult relevant literature. I see such occurrences as opportunities to uncover new mechanisms or refine existing theories. Documenting everything systematically ensures transparency and allows for constructive team discussions to reassess our research direction. 7. What statistical tools do you use in your research? I frequently use R and GraphPad Prism for data analysis. I'm proficient in using statistical tests like ANOVA, t-tests, regression analysis, and Kaplan-Meier survival curves. I also utilize bioinformatics tools for large-scale data analysis, particularly in genomics and proteomics. These tools help ensure the reliability and reproducibility of our results, enabling us to draw valid conclusions and publish findings in peer-reviewed journals. 8. Describe your experience working with clinical samples. I've handled clinical samples in collaboration with hospital partners, ensuring strict adherence to ethical guidelines and biosafety protocols. My role involved processing tissue biopsies, isolating nucleic acids, and performing histological analyses. I maintained meticulous records and ensured timely communication with clinical teams. Working with clinical specimens gave me deeper insight into patient variability and emphasized the translational value of research in bridging lab findings to real-world clinical settings. 9. How do you ensure ethical compliance in your research? I strictly adhere to Institutional Review Board, IRB, protocols and ensure informed consent when working with human samples. I follow guidelines from organizations like the NIH and IACUC for animal studies. I maintain confidentiality, data security, and proper sample handling procedures. Regular training on research ethics and compliance helps me stay updated and fosters a responsible research environment that prioritizes patient and subject safety. 10. What is your experience with grant writing and funding proposals? I have contributed to several grant proposals, including NIH R01 applications and institutional research grants. My role involved drafting research objectives, outlining methodologies, and budgeting. I've also led smaller grant applications for pilot studies. These experiences taught me how to align research goals with funding priorities and clearly communicate the project's significance and feasibility. Securing funding is crucial in cancer research, and I continuously hone my grant writing skills. 11. How do you manage your time during long-term research projects? I break the project into manageable phases with specific milestones and timelines. I prioritize tasks using project management tools like Trello or Microsoft Planner and set weekly goals. I also allocate time for literature review, lab work, and data analysis. Regular meetings with the team help track progress and address any roadblocks. This structured approach ensures steady progress and timely completion of research objectives. 12. How do you handle collaboration with other researchers or institutions? I believe collaboration enhances the quality of research. I maintain open communication, set clear expectations, and respect each team member's expertise. Whether working with pathologists, bioinformaticians, or clinical teams, I ensure data sharing is secure and timely. I've co-authored multi-institutional studies and coordinated sample exchanges, all while adhering to compliance standards. Good collaboration fosters innovation and accelerates scientific discovery. 13. What are tumor suppressor genes and why are they important? Tumor suppressor genes are essential in regulating cell growth and ensuring genomic stability. When these genes are inactivated or mutated, cells may divide uncontrollably, leading to tumor formation. Examples include TP53, BRCA1, and RB1. Understanding these genes helps identify mechanisms of cancer development and potential therapeutic targets. Researching tumor suppressors is crucial for early diagnosis, prognosis, and the development of targeted cancer therapies. 14. How would you explain your research to a non-scientific audience? I simplify technical terms and focus on the broader impact. For example, I might say, my research focuses on understanding how certain genes contribute to cancer and how we can stop them from making tumors grow. By studying these genes, we aim to develop better treatments that are more effective and less harmful. Using relatable analogies and avoiding jargon helps engage the audience and highlight the research's relevance. 15. What role does bioinformatics play in your research? Bioinformatics is integral for analyzing large datasets from next-generation sequencing and gene expression profiling. I use it to identify mutations, gene signatures, and molecular pathways involved in cancer. It helps us interpret experimental results, find patterns, and generate hypotheses for further validation. Collaborating with bioinformaticians enhances data accuracy and efficiency, ultimately accelerating discovery and enabling precision medicine approaches in cancer treatment. 16. What is your experience with animal models in cancer research? I have used mouse xenograft and genetically engineered models to study tumor growth and drug responses. I adhere strictly to animal welfare guidelines and obtain all necessary ethical approvals. These models allow us to study cancer biology in a living organism, providing insights that in vitro systems cannot. I ensure proper handling, documentation, and analysis to produce reliable and ethically sound data. 17. How you deal with research setbacks or failed experiments. Setbacks are part of the scientific process. When an experiment fails, I assess each step to identify potential errors or overlooked variables. I review literature, consult with colleagues, and modify the protocol if needed. I also maintain detailed lab notes for troubleshooting. Each failure offers a learning opportunity, helping refine future approaches, and building resilience and problem-solving skills. 18. Describe your publication history. I have co-authored several peer-reviewed publications focusing on molecular mechanisms in cancer and novel therapeutic approaches. My work has been featured in journals such as Cancer Letters and Oncotarget. I have experience with manuscript preparation, including data visualization, literature review, and responding to reviewer comments. Publishing is essential to share findings, gain recognition, and contribute to the global scientific community. 19. How do you approach hypothesis formulation in research? I base hypotheses on thorough literature reviews, preliminary data, and current knowledge gaps. I ensure they are specific, testable, and aligned with the research goals. Formulating a clear hypothesis helps structure experiments and interpret results effectively. I also consider potential alternative outcomes and design experiments that can validate or challenge a hypothesis through objective, reproducible data. 20. What is your experience with immunotherapy research? I participated in projects evaluating immune checkpoint inhibitors and CAR T-cell therapy in cancer treatment. My work involves studying how tumors evade immune detection and testing combination therapies to enhance immune response. Immunotherapy is a promising field, and understanding the tumor microenvironment and immune modulation is key to improving outcomes. I stay updated with the latest trials and breakthroughs to guide my research. 21. How do you document your research findings? I maintain detailed electronic lab notebooks, recording all experimental designs, observations, and outcomes. I use standardized templates for consistency and ensure data is backed up regularly. I also prepare progress reports and share summaries with team members. Good documentation ensures transparency, reproducibility, and facilitates smoother peer review and regulatory audits when publishing or applying for grants. 22. What are oncogenes and how do they influence cancer? Oncogenes are mutated or overexpressed genes that promote uncontrolled cell growth and division. When proto-oncogenes, which normally regulate cell growth, become oncogenes, they can initiate or accelerate tumor development. Examples include HER2, MYC, and RAS. Targeting oncogenes with specific inhibitors is a key strategy in modern cancer therapy. Understanding their function helps in designing precise interventions that minimize harm to healthy cells. 23. Have you worked with CRISPR or other gene editing technologies? Yes, I've used CRISPR-Cas9 to knock out specific genes in cancer cell lines to study their role in tumor growth and drug resistance. I've also used it for gene correction experiments in preclinical studies. This technology enables precise genetic modifications, making it a powerful tool in functional genomics and therapeutic development. I follow strict validation protocols to ensure off-target effects are minimized. 24. What's your approach to reproducibility in experiments? I prioritize standardized protocols, proper controls, and detailed documentation. I replicate key experiments multiple times and encourage peer validation within the lab. Using calibrated equipment and reliable reagents also ensures consistency. I follow FAIR data principles: findable, accessible, interoperable, reproducible, and openly share data and methodologies to promote transparency and scientific integrity in cancer research. 25. How you manage large data sets in your research. I use tools like R, Python, and specialized bioinformatics platforms for data analysis and visualization. For storage, I rely on secure cloud-based systems with proper labeling and indexing. I organize data by project, experiment, and date, ensuring easy retrieval and collaboration. Regular backups and adherence to data management plans protect against data loss and facilitate future research and publications. 26. What motivates you to continue working in cancer research? The potential to improve patient lives through scientific discovery keeps me motivated. Each experiment brings us closer to better treatments or a cure. The complexity of cancer challenges me intellectually, and the collaborative, ever-evolving nature of the field inspires continuous learning. Knowing that my research may help extend or save lives gives me a profound sense of purpose and responsibility. 27. How do you ensure your research remains patient-focused? I stay connected with clinical collaborators and consider real-world applications of my findings. Attending tumor boards, Reviewing clinical case studies and participating in translational research initiatives help me align lab work with patient needs. I also advocate for patient involvement in research planning, ensuring our goals reflect pressing clinical challenges and outcomes that truly matter. 28. How do you contribute to team projects? I contribute by sharing expertise, taking initiative, and ensuring open communication. Whether it's designing experiments, analyzing data, or writing manuscripts, I take ownership while supporting others' work. I'm proactive in troubleshooting and offer constructive feedback during team meetings. Respecting diverse perspectives and working toward common goals has helped me thrive in collaborative cancer research environments. 29. Describe a time when your research led to new insights. In a study on triple-negative breast cancer, we unexpectedly found a previously unreported RNA that influenced tumor aggressiveness. After validation, we proposed it as a potential therapeutic target. This led to a new research direction and a co-authored publication. Discovering something novel was exhilarating and reinforced the value of careful observation, persistence, and open-mindedness in the research process. 30. Where do you see the future of cancer research heading? The future lies in precision medicine, AI-driven diagnostics, and immunotherapy advancements. I foresee greater integration of genomics, proteomics, and single-cell analysis to tailor treatments. Liquid biopsies will likely become routine for early detection and monitoring. As we understand tumor heterogeneity better, personalized and less invasive therapies will emerge. Collaboration across disciplines will be key to accelerating breakthroughs and improving patient outcomes. Preparing for a cancer researcher interview requires both technical knowledge and a strong understanding of the field's broader impact. These 30 questions and answers offer a solid foundation to highlight your expertise, passion, and readiness for the role. Stay confident, stay informed, and continue striving to make a meaningful difference in cancer care.